Alright, what is going on, guys? We are starting the biophysics of psychedelic drugs. The reason I'm doing this is because I want to get into life sciences. I did graduate work in life sciences, not just physics, and so let's go ahead and get started. A few things first. There's a QR code to my Patreon page if you feel obliged to support my effort into bringing advanced science to the general public. Again, you don't have to. It's just there for your convenience. The other thing is that you can see that this is somewhat of a new structure of video, right? I have most of the screen now is the workspace. Excuse me. And also I'm recovering from a cold, so if I'm coughing a bunch, just ignore that. And then there's me up here in the corner, okay? So with all that out of the way, let's go ahead and get started. So we're talking about the biophysics of psychedelic drugs. And these drugs are... I chose this topic because it's interesting, right? A lot of people find this interesting, but it's also technical. And when you're out there, you're looking for a job, especially in the life sciences, which is kind of hard now, I feel like. There's... the employers want you to know some technical stuff, right? And this series is going to help you in understanding some technical stuff when it comes to, we'll learn how to purify, we'll learn the whole process of purification, we'll learn computational stuff, we'll learn so many things here. We're going to learn some structural biology, we're going to some electrophysiology, we're going to learn a lot of this technical stuff so that you can maybe put this on your resume and say, you're somewhat fluent, at least, in understanding how to do understand basic chemistry. And I'm doing this in sort of the guise of psychedelic drugs because they're interesting. They're all over YouTube. They're all over common culture and all this. And so, yeah. So let's go ahead and, I've said get started already. Now let's go ahead and get started. So I mentioned really briefly the cultural relevance. What we want to do in this video is really just introduce the topic, okay? The idea here is I actually want a video to come out every day. And so this is going to be somewhat of a task. Here, I'll erase this really quick. This is going to be somewhat of a task to do, but I think I'll be able to do this. So, and if not, there might be 1 or 2 day gaps in between some videos, but that's all in me, right? So, in pop culture, we have, if you're an American, I know some of my audience is Indian. I don't know if you'll know this then, but Matthew Perry, a famous actor from the show Friends back in the 90s, is known to have died from a ketamine overdose. And ketamine is a psychoactive compound. It's not hallucinogenic. That's a big common misconception. I feel like a lot of people don't understand that psychoactive doesn't equal hallucinogenic. And we'll talk about that in a bit here. But ketamine is considered a psychoactive compound. And ketamine is known, as far as we know right now, to have killed Matthew Perry. Okay, he overdosed on ketamine. And then there's this relevance in religious culture. Excuse me. In religious culture, we have this very, very interesting body of work that has emerged over the past, 50 years or so, on how mushrooms in particular, but many plants, have played a role in the evolution of religions. And if you read the Bible, which I have done, there's this wood called shittim wood, Shittim wood. I erased it here. I can write it down again. This is from the acacia tree, and this is a natural producer of something very analogous to DMT. I don't think it's exactly the same, but it's very, very analogous. And DMT, if you are aware, is naturally produced also in the pineal glands back here. Like the pineal glands are somewhat back, like right around your ears, like right behind your ears. There are these little glands that release DMT naturally when you're sleeping. And when you're sleeping, these that's what causes you to hallucinate, right? The DMT is what causes you to hallucinate, and you're dreaming, right? So a dream is a hallucination because you're seeing things that aren't there, and you're dreaming, right? So this is that's how DMT works, right? And the acacia tree and the shittim wood is mentioned many, many times in the Bible, and this is this kind of stuff is a hallucinogenic compound. And so we have these books, right? The Psychedelic Gospels, Sacred Mushroom and the Cross, Food of the Gods. Food of the Gods is a particularly famous one, I think, by Terence McKenna. But the creme de la creme, if you will, the bad boy of all these books is going to be The Doors of Perception. The Doors of Perception is a book written by Aldous Huxley. He's an English writer and philosopher. And he decided to take about 5 mg of mescaline, dissolve that in water, take it, and then record his experience. And mescaline is also a hallucinogenic compound. It's psychoactive, and excuse me, I'm sort of on the mends of this cold here. What was I saying? So the psychedelic compounds are, or the mescaline experience is what he wrote about, and then he codified that into this book, which was The Doors of Perception. So that's this book right here. Right, so, and this is like a must-read book if you're into this kind of stuff, right? It's really short. It's like not even 100 pages, I don't think. But super, super interesting, super relevant to what we're talking about. All right, speaking about The Doors of Perception, I recently watched a very recent video that came out on YouTube with Alex O'Connor. I think it was Dr. K, and some theologian, I don't really know who he was. But let's watch Alex O'Connor talk about The Doors of Perception, because I think this is somewhat relevant as well. Let's see if I can...
Brain, as Aldous Huxley said, was a tool for focusing the mind. Aldous Huxley writes in the Doors of Perception. Essential reading to anybody interested in consciousness, by the way. Aldous Huxley in the 20th century takes a psychedelic drug, and he writes about his experience, and he writes about it beautifully. And one of the things he realizes is that his mind has been opened. And he thinks, okay, well, if my mind has been opened during this experience, then that means that in normal waking hours, something must be closing my mind. What could be closing my mind? Answer: the brain. He concludes that the brain is a tool for focusing the mind. So, the psychedelic experience, this is before we've done any scientific experiments on this. You can scan people's brains in like an fMRI scanner, right? Okay. So, when you take a psychedelic drug, your experience just blows up, right? You start seeing colors you didn't know existed. You start experiencing things as if they were new. It's like the experience is unimaginable. So we've taken people and we've measured their brain activity, and their brain activity is at a certain level. And then you give them a psychedelic drug and you put them in the MRI scanner, and their brain activity goes down. Brain activity goes down as the mental experience expands and goes up, which for the person who experiences the psychedelic drug, they will report this as a feeling that they just get from the experience. The scientist who measures the brain activity, the sages writing the Upanishads, the Buddhist monk after a series of long meditation will all say the same thing, which is that in some inexplicable way, consciousness is more foundational than the brain is. And the brain is focusing consciousness, and in some sense that means that our individuated selves are essentially illusory.
So there's Alex talking about the effect that psychoactive compounds or psychedelics have on the consciousness, which is super, super interesting. And as I was going through my graduate work, I thought, I was talking to someone once, and they told me once that the brain is primarily an inhibitory thing, right? So when it's active, it's the GABA receptors, in other words, are the things that are mostly at work in the brain. And as you decrease that activity, right, it becomes more excitatory, which is kind of intuitive, counterintuitive, if you will. And so but it sort of makes sense within the light of what Alex is talking about. And we'll talk about another book here, which is the Origin of Consciousness and the Breakdown of the Bicameral Mind by Julian Jaynes. I don't think Alex has read this book, but it talks about sort of the same thing, how specifically in, I think it's chapter 3 of the book, it talks about the neuroscience behind paranoid schizophrenics, people who are seeing things that aren't there, but nevertheless are acting as if they are there. And in the book, it's very interesting. It argues that something very similar to what Alex is saying. It argues that our ancient ancestors, in some sense, they had more of this. I wouldn't say reptilian brain, but they had this sense of consciousness that was a little bit different than what we have nowadays, where on one half of the brain they have this linguistic sort of analog to Wernicke's area in the brain that communicates to the other side of the brain, and as it's communicating in the form of dictations, and his hypothesis in the book is that these dictations are the dictations of gods, right? And so consciousness, that's what consciousness was, sort of, according to James, right? And as time evolved, as globalization happened, as more giant natural disasters occurred, the volcano on Crete, or I think it was Crete or Knona, an island off of Greece, as all of this happened, this forced populations to sort of spread out and then come in contact with more populations over time, forcing the brain to shift this form of consciousness into a new form of consciousness, which I think is super, super interesting. So let's not dwell on this too much. I want to look at one more video on Jordan Peterson talking with Matt Dillahunty about the effect of psychoactive chemicals, because this is going to put us into a very interesting domain on how this is all relevant to biotech, biopharmaceutical companies, and all of this. So let's take a look at this.
There's good reason to believe there's God. That is where the focus is primarily.
Yeah, and it's usually construed as a scientific issue.
Well, it's not so. I would never construe it as a scientific issue, because science can't confirm the supernatural. I mean, we are blocked until somebody demonstrates a mechanism by which we can explore and confirm the existence of the supernatural, and then confirm that it actually can impact the natural world. We're stuck, which is why science relies on methodological naturalism. You
seem to be able to do it with psychedelic drugs.
You seem to be able to do what with psychedelic drugs?
Well, everybody that takes them under reliable settings generally comes back and claims the presence of a mystical experience.
Yeah,
and that seems to be duplicatable and replicable across cultures in labs with multiple different substances, the chemical constituents of which are known very well. It's part of this religious phenomenology. And to me, that's just a set of facts. And we know that people have been using those substances to evoke mystical and religious experiences for, who knows, 150,000 years maybe. Sure. We can at least infer that. And we have no idea whatsoever how much of an effect the use of those substances has had on the metaphysical substrate of our culture. So what constitutes scientific data here is by no means clear. Here's an example of this, for example.
Before the example,
yeah?
As someone who's been high as a kite, I fully agree that people take drugs and report experiences that they describe as mystical or supernatural. Just like people report other experiences as if they were religious or ghosts or whatever. We have no way of confirming that this something mystical or supernatural actually happened. What this is about the language.
Stops people from smoking.
Well, you can stop smoking without any sort of supernatural intervention.
No, not really.
You can't stop smoking without supernatural?
There aren't really any reliable chemical means for inducing smoking cessation. You can use a drug called bupropion. I think that's the one. It's whatever Wellbutrin is.
Is that supernatural?
No, you don't need a supernatural effect, but it doesn't work very well. But if you give people magic mushrooms, psilocybin, and they have a mystical experience, they have about an 85% chance of smoking cessation with one treatment. But that's kind of like evidence, It's kind of like evidence.
It's
And
evidence that you can take mushrooms and increase your chance of quitting smoking. But
no, it's not. It's indication that if you take mushrooms and you have a mystical experience, you'll stop smoking, because it doesn't work if you don't have
if
the
you take
experience.
mushrooms and you have an experience that you describe as mystical, then you'll increase your chance of smoking. But that doesn't tell me whether or not there was something to this notion that they had an experience that was supernatural in any sense.
Well, it's not definitive evidence.
It's
but
not evidence at all.
Oh, sure it is. Oh, sure it is.
No,
How is it? Wait a second. Wait a second. That's wrong.
Okay.
It is evidence. So, because look
no, he's right.
You want to speak,
He's right.
you want to think
I
through
will
this
correct that.
skeptically. Okay, you have a pharmacological substance, it's psilocybin. You give it to people who are trying to quit smoking. The psilocybin doesn't directly have an impact on the smoking behavior. It has to elicit what's described subjectively as a mystical experience. And you can get physiological indicators of that mystical experience. And you might say, well, that's not enough to prove that it's a mystical experience, but you're conscious and I accept that. It's like you accept all sorts of things without being able to demonstrate their validity on every possible objective with every possible objective criteria. So don't get in too much of a hurry. It's a serious issue. If you give people psilocybin, for example, and they have a mystical experience, not only are they much more likely to quit smoking, which is really something, but they're also much less likely to suffer from death anxiety if they have cancer. Like, that's quite the thing. And not only that, if you test them a year later and they've had a mystical experience, which the majority of them regard as the most significant one, two, or three experiences of their life, including such things as getting married, their personalities are permanently altered in the direction of more openness to experience and more creativity by a standard deviation. Like, that's walloping effects. So we can't get too much in a hurry about dispensing with all that.
All right. So what Peterson is talking about here, I think, is super, super interesting. And it gives credence to sort of clinical evidence as to why I think psychedelics work, or why psychedelics should be taken seriously. And so was Jordan Peterson right? We should always be skeptical about these claims. So I think he actually was. If you look at the research and the articles, I brought up 2 articles here. Was Jordan Peterson right? Well, in John Hopkins, there was results excuse me, of psilocybin. Again, psilocybin was the main chemical that was being used here. At 6-month follow-up, 80% of participants had achieved 7 consecutive days of abstinence from smoking, confirmed through both self-reports and through biochemical verification. This is super, super interesting. To put this into perspective, the most effective traditional smoking cessation treatments typically achieve rates of 15 to 35%, and this was 80% after taking psilocybin, right? So that is extraordinary. Now, if we look at this other paper that I found, this here says their findings. I'm not going to go through the entire methodology here. This is a randomized clinical trial of 24 participants with major depressive disorder. So this is treating depression, not smoking cessation. Participants who received immediate psilocybin-assisted therapy compared with delayed treatment showed improvement in blind clinician rate-assisted depression severity and in self-reported secondary outcomes through the 1-month follow-up. So psilocybin helps with depression as well. And that leads us into this next topic on why psychedelics, I think, are very, very relevant within the biotech and the pharmaceutical industry. So AbbVie, which is a pharmaceutical company, tunes into Gilgamesh's story. If you're not aware of what Gilgamesh is, it's just a very old, ancient story. I'm not going to get into it, but with Enkidu and it's a very cute little story, and I think it's humanity's first story. You can correct me if I'm wrong on that, but inking $1.2 billion deal for a psychedelic program. So they bought out $1.2 billion worth in a psychedelic program, right? That is incredibly interesting, right? This idea that psychedelics are sort of becoming mainstream in biotech. I say becoming mainstream, that leads us into the next slide here, which is relevance to depression, right? So psychedelics are going mainstream. Are you ready for it? This was a talk that I actually attended a while ago. If we go back actually really quick to this one, this was in 2020 or 2025, this year of August, so really not that long ago. And then there's this one here. This was back in June of 2025, of which, again, I attended. I thought it was super, super interesting. They're talking about psilocybin. It's used in curing depression. Synthetic, or, excuse me, my gosh, a version of LSD that was, I think, called MM60 or something like that, if I remember correctly, that was also being used in clinical trials as well. So here we have it. We have the relevance to this kind of stuff. Super, super interesting. Now, let's make a clear distinction here. As I've noted, psychedelics do not equal hallucinogenics. Okay? Psychoactive is a broad, overarching category, and hallucinogenic is a specific subcategory within it. With a psychoactive compound or a psychotropic substance is any chemical substance that acts primarily upon the central nervous system, the CNS, to alter one's psychological functioning. Okay, and now here are some examples. We have depressants, right? We have depressants, we have stimulants, we have hallucinogens, and we have antidepressants. These are all different drug classes. These are the four main drug classes. And there's their effect on the central nervous system. Depressants will slow down, stimulants will speed up, hallucinogenics will cause major alterations in perception, as you probably are well aware of, and antidepressants modify mood cognition for therapeutic purposes. And examples for depressants include alcohol, benzodiazepines. Again, benzos are, I think, what Jordan Peterson was taking, opioids, and fentanyl. Well, we're going to talk about fentanyl in the next video and talk about specifically some biophysics as to why it's really, really bad. Okay. And then we have, for stimulants, caffeine, nicotine, cocaine, amphetamines. And we have, for hallucinogens, we have LSD, psilocybin, ketamine. And, ketamine is a hallucinogenic. Okay. I might have said that it wasn't when I was talking about Matthew Perry. And then antidepressants, which is SSRIs, Prozac, and TCA. Right, so just some examples, right, in the drug classes. But notice how hallucinogenics are within these drug classes, right? So this is a subgroup of psychoactive compounds. All of these are psychoactive compounds, right? All right. So the next thing we want to talk about is, where do these drugs come from? Well, many of them are just naturally produced by plants, right? So we have the peyote, right? So this here is peyote. I think that's how you spell it. This is muscimol. This comes from the Amanita muscaria mushroom, which is, I think, native to the Amazonian jungle, on like the eastern ends of Peru and western ends of Brazil. Shout out to my Peruvian cousins, right? Part of my heritage is from there. And then we have cannabinoids, right? This is a weed, right? And then we have DMT, right? So DMT, right? So DMT is that's the thing that's naturally produced by your brain, right? So this is all super, super interesting stuff. But then we have, this is LSD. LSD, this was made by a guy named Albert Hoffman. This is synthetic, right? So some of this stuff is going to be synthetic. Some of this stuff is going to come from plants and mushrooms. Mushrooms, I think, are the sort of the well-known things that create psychoactive compounds and primarily hallucinogenics, I think. But yeah, so these are just some examples. I want to also talk about a few books that I would totally recommend if you're into this stuff. So let's look at these books really quick. The Doors of Perception, we talked about. The Origins of Consciousness and the Breakdown of the Bicameral Mind. Again, something that I also mentioned. And the entheogens and the development of culture. This is interesting. There's a quote I have here. Researchers suggest that we have purposely enhanced receptor sites in the brain, especially those for dopamine and serotonin, through the use of plants and fungi over a long period of time. So this almost seems to be going against the grain of evolution, if you think about it, right? This is this idea that evolution is this thing that nature imposes constraints on organisms, boundary conditions, if you will, on organisms, which shapes their phenotypic, what shapes their phenotype, right? It shapes how they function in the world. It shapes how the genes sort of manifest themselves in the world. But the willingness to take a plant, like peyote or something like that, is not really nature putting constraints on us. It's us sort of opening our horizons, if you will, our visual or our psychoactive horizons, in a way that fundamentally altered our brain chemistry and perhaps even our brain genetics. So, because again, the serotonin receptors, they come from ultimately the genes and the nerve in the nuclei of the nerves in our brain. So there's that, right? So I would totally recommend these 3 books if this is something that you're really, really interested in. So let's go ahead and start getting into some of the neuroscience to get this out of the way. And then we will start talking about some of the physics involved, right? So how do neurons work? Okay, well, you have a presynaptic neuron. I'll use my hands here. And you have a postsynaptic neuron, right? So the presynaptic neuron is going to send signals, right? It's going to send an action potential down its axon, and it's going to meet at something called a synapse. And at that synapse, something happens, right? And then the action potential is generated again down the next neuron. Okay. What happens at the synapse is super, super important. So calcium permeable receptors allow for vesicles filled with neurotransmitter to attach to the cell membrane to release the neurotransmitter at the synapse. All right, so that's going to be these guys. They're on the postsynaptic side, or not these guys. That's going to be it's going to be these guys, right? Because they're calcium receptors, they're not attached to G-alpha proteins, but that's a small detail. Vesicles containing neurotransmitter are shuttled towards the synapse via a protein called kinesin. And then when they get here, the calcium from these guys is going to aid in little proteins right here attaching to the vesicle and then pulling the vesicle and opening it up, releasing all of its contents into the synapse. And then that content, the neurotransmitter, is going to bind to receptors on the postsynaptic side. Right? So there's 2 pathways that happen, 2 main pathways that happen when a cell receives a signal like this. Okay? The first main pathway is ions, right? Ions are going to flow into the cell, right? When the neurotransmitter binds to the receptor, it opens them, ions will flow in, and that's going to create another action potential. Okay, so that's the action potential route, right? And then there's going to be the cell signaling route, right? So neurotransmitters will bind to a receptor. It doesn't open, it like moves and bends and folds in some weird way that causes it to be more that causes a subunit within the cell to break off, or for it to become more have a higher affinity for another protein inside the cell to bind to it. This is all of that is cell signaling. Okay, we're not going to go too deep into cell signaling, but that is so two things happen. One is the generation of an electrical action potential, okay, and the other is stuff inside the cell starts doing stuff, right? Those are the two main pathways, okay? All right, so what does this mean? Well, if we zoom in on the synapse a little bit more, this is super, super interesting. This took me quite some time to make, but here we have our two pathways, right? So neurotransmitter in the vesicle is going to fill the synapse. It's going to bind to a receptor here. Excuse me. And then ions, right? These little ions are going to flow through the receptor, generate action potential. So that's one way that nerve cells are going to do this. You probably know this already if you know a thing or two about physiology. This is just review and an introduction. And then there is going to be the method where you have neurotransmitter again. So neurotransmitter is going to bind to this protein, right? This protein's weird looking. It doesn't open, but it bends and folds and changes in some way. What's called a conformational change, right? So I'll write that down actually. Conformational change, right? And when it does that, that starts to induce secondary messengers like CAMP and all the rest, like PKA and phosphorylation activity, and it's like a domino effect, right? That's what happens. So you have this action potential and this domino effect of proteins. Those are the two main pathways that happen. And we're going to talk about something called a non-canonical pathway in a bit. But let's take a look at the action potential, because again, we just want to get a lot of this introductory stuff out of the way. So when we're talking about action potential, again, the cell likes to stay polarized. Let's just assume that the cell likes to stay polarized. And so it's kind of like society, right? We live in a very polarized society. It's always been somewhat polarized. Society nowadays seems to be a little bit more hyperpolarized due to politics or whatever. But cells are kind of like society, right? They like to be polarized. Or they tend to be polarized. I wouldn't say like to be polarized. They tend to be polarized. And the depolarization process is when ions flow into the cell. So you have the neurotransmitter binding ion, positive charged ions will flow into the cell. Okay, and then as they're flowing into the cell, depolarization happens, right? So you have society, the cell, it's polarized, and then ions will flow in, becomes depolarized, and then new channels will open, right? Allowing for potassium inside the cell, which is positively charged as well, to leave, right? So you have a flow of sodium into the cell, and then you have a flow of potassium outside of the cell, okay? And that's going, when you have positive charge leaving, the inside becomes negative, and you get that repolarization. So it's always polarized in the negative direction, right? These cells are always polarized at -70. But then this repolarization is going to go all the way down. It's going to be a little bit too polarized now, right? This is going to be like the society we're in right now, right? Where the politics or whatever, it's just where it's way, way too polarized. So it has to then depolarize a little bit more, and that is done through these receptors, or these channels, or receptors, if you will. They're not really receptors, but these proteins here, right? These are going to, the hyperpolarization occurs when the net charge inside the cell is negative. At this point, the cell depends on sodium-potassium pumps to reestablish sodium on the outside of the cell and potassium on the inside, right? So sodium went in, potassium goes out. That going out is the repolarization, but becomes too polarized. And so we need pumps that that reestablish that gradient. It brings the potassium back in, and it forces the sodium back out, right? So that is how this works. That's how the action potential works, right? So it's just you can almost think of it as like this dynamic interplay between sodium and potassium. Potassium's inside the cell, sodium's outside the cell, right? Sodium's going to rush in, potassium's going to rush out, and then to reestablish that gradient, you need this sodium-potassium pump, okay? All right, and then myelin sheaths are going to speed up the process, right? So action potentials are unidirectional. Myelin is an insulating layer that forms around the nerves. The myelin allows for a jumping of the action potential down the axon so that the action potentials can travel much faster. All right, we're not going to get too much into that here, but this is just a brief rundown on action potential. Okay, so the next two slides I'm quite proud of. Before I get into them, we want to talk about these receptors, right? So these receptors, you might ask, okay, how do we know what these receptors look like, right? Because these are going to be the ultimate targets of the psychoactive compounds that we're talking about. These are going to be the what we're really, really going to be interested in. And when I was doing my graduate work, I was working on glutamate receptors, right? So, and I was doing cryo-EM on glutamate receptors. Cryo-EM is super, super interesting. Now let's take a look at cryo-EM structures of some of the most relevant receptors that these psychoactive compounds are going to attach to. We have kainate receptors. Cryo-EM is such an interesting thing. You get maps of proteins like this, and it's just so cool to see, right? It's colorful. Well, the colors are made after the fact, but you have the kainate receptors, right? So kainate receptors, ketamine is something that is known to bind to kainate receptors, and these are a type of glutamate receptor, right? So glutamate receptor. NMDA receptors are also well known, or they're more well known. These are also glutamate receptors. And AMPA receptors are also glutamate receptors. There's also something called the GLU-D receptor, a little bit less well known. Kainate receptors are a little bit less well known as well. But then we have the big famous serotonin, that's the 5-HT3A, right? Those are the those proteins. We have GABA, right? GABA is well known. That's an inhibitor. That's an inhibitory receptor, right? That allows for negative ions to come through. And then we have and then we have nicotinic acetylcholine receptors, and glycine receptors, and these are all classes of something called, or types of something called ionotropic receptors. So if we go back really quick to this slide, this is going to be the receptors that generate the action potential, right? They allow ions to flow through them. They're ionotropic, right? They're the things that allow the ions to pass through them. And you could sort of see, like, you can sort of infer that, yeah, maybe, like they look like these things that maybe have a center to them that ions will run through. But okay, so let's take a look at metabotropic receptors. So metabotropic receptors are receptors that do the conformational changes and then induce some sort of secondary messenger system in the cell. So we have dopamine receptors, right? So dopamine is when you go into YouTube, for example, and you want to quench your tribalistic instincts by, I don't know, listening to someone you hate. Like that dopamine's involved there, right? Serotonin receptors are also have a metabotropic form. Glutamate receptors also have a metabotropic form. Acetylcholine receptors also have a metabotropic form as well. And this is not this is not a canonical list. There's more of these receptors out there. I've just taken some of the most common, well-known ones. We have adrenergic receptors, right? And GABA receptors also have a metabotropic form as well. You can see, when you look at these things, like the outside of the cell is this part here, right? So there's not much there. There's going to be a binding site. There's going to be a small binding site for the neurotransmitters to bind, but most of the bulk of the protein on the inside is going to be that thing that bends, moves, and changes, and then it has it increases its affinity for pKa or whatever to induce those secondary messengers. Adrenergic, and then we had the cannabinoid receptors, right? So if you're taking weed, for example, or cannabis, right? Cannabinoid, cannabis, that is going to affect these receptors, right? These receptors are part of something called the endocannabinoid system. Endocannabinoid system. And there's all these different systems, like sort of subsystems in the brain that I don't think a lot of people are very aware of, right? One is the endocannabinoid system. There's the glymphatic system. There's the limbic system. Limbic system is a little bit more well known. But so we have these metabotropic receptors, right? That's the point I'm trying to get across. Okay. Usually when you read the literature, if it's not cryo-EM, these receptors are going to look a little bit more like this probably, where you don't have you can't see all the alpha helices, but they're just cartoons. And I've just taken a class to illustrate here the incredible diversity of these proteins. I've just illustrated some of them here, right? So we have NMDA receptors are a class of glutamate receptors, as I've said before. AMPA receptors, kainate receptors, GLUD receptors. We have the metabotropic forms as well. But what's interesting, if we go back to here really quick, what's interesting, let's take a look at the kainate receptor, for example. I'll erase some of this stuff. We have a blue part here. We have an orange part here. And we have a red part here. These are all, the blue and the orange part are subunits, right? The red we can kind of ignore. Those are different proteins that interact with the kainate receptors. But the blue and the orange, those are subunits. The blue is going to be something like a subunit 2, and the orange is going to be something like subunit 5 or something like that. So this could be, and normally when this is like subunit 2 and this is subunit 5, the way they're distinguished is through their sequence, their amino acid sequence. And they can only combine in certain ways, right? They're called like glucate. You'll have something like GluK2, right? That's glutamate receptor, kainate receptor 2. That 2 refers to the unique sequence, the unique primary amino acid sequence. So you have GluK2, you have things like GluK1, right? And this is going to run through a different set of numbers. And when we look at this, again, for example, we have GluK1 to 5. So there's 5 different subunits. That red thing that we saw in the cryo-EM structure, that was something called a netto protein. Those proteins will bind to the glutamate receptors, and they will sort of modulate their function, right? So the glucate-2,5, for example, that's going to be like blue and orange. That's going to have some function to it. But when you have these other proteins start interacting with it, that function is going to get modified. And we'll see how function in later videos gets measured by these proteins. And then we have the metabotropics, right? So the metabotropics also have their incredible diversity as well. And this is fundamentally the diversity comes in the form of the amino acid sequence, right? All right, so we're almost done with this section here. I don't want to belabor this too long, but let's take a look at the psychoactive chemicals themselves now, and some of the most well-known ones, right? So this is going to be psilocybin. We have muscimol. This comes from the Amanita muscaria mushroom. Then we have LSD. This is lysergic acid, diethylamide. We have ketamine, right? And then we have 3,4-methylene dioxymethamphetamine. That's MDMA or ecstasy. And then we have mescaline or peyote. And then we have the tetrahydrocannabinol, and that's THC or cannabis, right? And these things have three-dimensional structure to them. They're not these two-dimensional things. The bonds can rotate, right? If you have a single bond, if you've taken chemistry before, the bonds can rotate, right? If they're double bonds, they don't really rotate. They have oxygen, right? So red is oxygen. I can actually do this in red because why the hell not? I think I might have to do it like this. Why did that not go red? Try something here really quick. That's weird. I wonder why my pen isn't going. Oh, there we go. So oxygen, all right, and then we can do blue, right? Blue is going to be nitrogen, and then we can do green. Green is sulfur. All right, so if you're taking chemistry, that's what I'm doing here, right? I'm just identifying what the colors are. And then we have these dots here. Those dots that represents aromaticity in the structure. If you don't know what aromaticity is, it just it's a type of resonance, right? Again, I'm not going to get too much into the general chemistry here. The idea here is that we want to go down a very specific route, looking at the biophysics of drugs. Along the way, though, we'll touch upon some general topics as well, so that you can learn the general, but you can also, on the way. So you're learning the general, but on the way, we're heading towards something very specific. All right. And last but not least, I want to also mention this non-canonical pathway that I had mentioned before. Now, this is interesting, right? So you have nerve cells, right? These nerve cells send action potential, meet at an axon, and then send action potential, right? So there's this sort of lateral communication, right? But there's also horizontal communication as well. And the way this happens is you have these, so you have glutamate, right? So glutamate is going to get released. Some of this is going to diffuse, though, right? Just by natural diffusion. And what's actually very, very interesting, specifically about kainate receptors, if you look into the literature here, the kainate receptors, is that they actually serve as metabotropic, right? Even though they're ionotropic, they actually have a metabotropic function if they are on the presynaptic side of GABA-releasing terminals. So glutamate, a glutamate nerve cell, not only talks with the subsequent nerve cell that it's sharing a synapse with, but it's also talking with neighboring cells, right? So this is super, super interesting. And this talking is going to modulate or change how GABA is released, right? This is super, super, super interesting. And I would, if I was you, I would totally start reading up on this. When I was doing my graduate work, when I started reading up on this, I thought, wow. It took me a while to understand this. This was like lateral communication. And I thought this is like super, super interesting. But yeah, so most of the neurotransmitter is going to bind to these guys, right? Because there's less of a distance involved. So if the distance is lower, then then it's going to be faster to bind because the affinity is just so much higher there as well. But there's going to be a non-zero probability or a non-zero chance that this happens also. All right, so to give credit where credit is due, for all of these videos, I'm going to put references. All of these references are for the cryo-EM structures that I'm putting in this playlist. Shout out to this guy right here, if you're watching, Kevin Felt. I know him. He's a cool guy. And so with that being said, make sure to hit that like and subscribe button. Make sure to hit up my QR code as well if you feel so inclined to do that. And I'll see you guys in the next video.